Okay, so I want to go back a little bit to more classical techniques. So from a panel of biochemical tests or Maudit-Toff spectrum, you can identify bacteria down to the species level. But often it's very useful to have subspecies identification because, for example, if you think about Escherichia coli, most of the isolates of E. coli are harmless commensal bacteria, but there are several types of pathogenic E. coli. So if you've got an identification E. coli at the species level that you've isolated from somebody, you might want to know whether it's a pathogen or just a commensal strain. And that's also the case for quite a lot of other opportunistic pathogens like Streptococcus pneumoniae. Now, to get subspecies identification, you can carry on with the kind of biochemical approach and test whether your bacterium can ferment a particular sugar perhaps, but mostly for subspecies identification, serological techniques are very, very useful. So within a species, you will have different serotypes of bacterium, and this will depend on the structure of the capsule antigens, flagellum, present or not present. If it's present, what is the serotype? And serotype of the O repeats in LPS. Now, serotyping is often done, okay, it's just going to hang there, by an agglutination test, which is very quick, and I'll just draw you how it works on the board here. So, for example, if you've got a bunch of bacteria that are, I don't know, 01157. This is E. coli. The LPS O antigen is serotype 0157. You suspect this. So what you do is you isolate, or rather mix, these bacteria with some latex beads that are conjugated with antibodies specific for this antigen. So you have these bacteria from colony on a plate. You make a suspension in water or saline solution. Mix the bacteria in suspension with the beads. And what will happen is that the beads or the antibodies on the beads will bind onto the bacteria, and this whole suspension will agglutinate. So instead of having beads in suspension, bacteria in suspension, the antigens recognize the antibodies, and you get a kind of big lump that is held together. So this is a very good test because it's quick. You incubate at room temperature, on the workbench for 20 minutes, and you get the result. You can see, maybe not by eye, but you look at the slide under a low power microscope, and you can see whether you have something where the beads and the bacteria remain in suspension. This is a negative result. Or whether you have clumps, agglutination. This is positive result. Okay, so depending on what antigen you want to detect, you'll use beads which have antibodies specific for different capsule hemagglutinin or O antigen serotypes. Okay. So that's often used for identifying different strains of pathogens. For example, if all of you were to come down with or let's say a bunch of students over the same couple of days all caught salmonella, and there was some salmonella isolated from the mayonnaise in the university restaurant, in the university canteen, then you might want to know whether it's the same bacterium in the people who got sick and in the dodgy mayonnaise. So in order to do that, you'd want to get the species identification and the subspecies identification. And this would generally be done by serotyping. Okay. So alongside that, there are a lot of molecular techniques that can be used. MLST, multiple locus sequence typing. So you amplify up a bunch of genes by PCR and sequence them. And then you can compare them. Genetic fingerprinting, these depend on the presence in bacteria of repeated sequences that are scattered around the genome. And their distribution is different between different isolates. So just with the primers that are specific for some of these repeated sequences, you get a different kind of profile depending on the strain of bacterium. And these are present in most gram-negatives and some gram-positives. So those are the techniques that you can use to identify bacteria. Now, identification is one thing, and how do we build taxonomies? Even why do we bother? Because one of the reasons is we want to know the evolutionary relationships between different bacteria, either at a low level. For example, if there is an outbreak of plague bacterium somewhere, or cholera, then you might want to know where this the outbreak came from. So it's useful to build a taxonomy to be able to identify the most likely or the nearest related strain. Okay, I don't know if you remember when there was the earthquake in Haiti a few years ago, right? And then there was a big international aid effort, which was followed by a cholera outbreak. And the cause of the cholera outbreak were some international, I think it was military personnel from Nepal, because there had been cholera circulating in Nepal just before. These people brought it with them, and in the kind of poor hygienic conditions of their camp, well, they sparked off the cholera epidemic in Haiti. And you were able to show this by kind of building the taxonomy and showing that, yeah, the strains that were circulating in Haiti, their nearest common ancestor was the strain circulating in Nepal. So constructing taxonomies, you know, it's an interesting exercise, but it also has a practical utility sometimes. Now, if we go back in time till when sequencing was expensive, and we're talking about 25 years ago here, or even more, and there was a kind of an effort to use the phenotypic information that came from morphology, serology, biochemical tests, and use this information to create phylogenies. So, you know, when I talk about a character here, okay, a character, this is whether the bacterium can ferment lactose or something like that. So that's it's yes or no. So if it can, then the bacteria, you say, okay, this bacterium, lactose positive, it's got 1. If you can't ferment lactose, then you'll have a 0. So each bacterial species that you have, for each character, you score them for 1 and 0. And then what you do is compare pairs of organisms, or let's say used to do, and you construct a table like this. So you've got the two taxons that you want to compare, two bacterial species. You count up all the characters that are present in both cases, the characters that are present in the first bacterium but not in the second, and those that are present in the second but not in the first. And in D, this is the number of characters that are absent in both. And from this table, you can calculate a matching coefficient. So the simple matching coefficient is everything that is a shared character, whether it's present in both or absent in both, divided by the total number of characters you have studied. Can anybody see a problem with this? Yeah. We'll see this coming up in a minute. That is one of the problems. Well, one of the things here is when you count characters that are absent from both taxons as having equal weight to when the character is present. This is poor practice phylogenetically, okay? Because lacking something is not really positive evidence of a very close phylogenetic relationship. So this is probably not a good idea to use this simple matching coefficient. It's probably better just to ignore the characters that are missing from both taxons in a pair and just consider the characteristics that are present in both taxons. So this makes more sense phylogenetically. So once you've done that, you'll have a number between 1 and 0, right? Whichever one of these coefficients you have. It's going to be somewhere between 1 and 0. 0, they don't share anything. 1, they're perfectly identical. And this will allow you to construct a distance matrix. Okay, so just like in first year when we did phylogenetics, and the closer the two taxons are, the closer this number will be to 1. So then once you've got this table, then you can use distance methods like NG, neighbor joining, to reconstruct phylogeny. So that's quite nice. It should work, right? Okay, you can see that taxon 4, it's far away from everybody, so it's the one that's furthest out on this phylogram here. 1 and 2, they're the closest together. Okay, so that's the technique for constructing phylogenies from characters, and it was applied to quite a different number of bacteria. So here's a paper I found from 1994, and the authors were interested in classifying different species of Neisseria. So Neisseria has got some important pathogens in the genus, Neisseria gonorrhea, Neisseria meningitidis, and also a number of harmless commensal bacteria like N. polysaccharia and Neisseria lactamica. You know, you'll probably be carrying some of these in your body, and they are perfectly harmless. So this was a phylogeny created with characteristics, and as you said, the number of characters you need to use is going to increase depending on the number of taxons you have to classify. So these are taxons up here, and they had to test all of these biochemical characteristics on all of these bacterial species. So it becomes really quite a lot of work to get this done. So anyway, they established this phylogeny based on phenotypic characteristics. So let's fast forward a couple of decades now. Sequencing is much cheaper. PCR is a pretty standard technique everywhere. And this means that molecular techniques are, in fact, much easier to use now than doing this kind of experiment. So the first thing that was done was really just to hybridize genomes. So you get the DNA from one species, label it radioactively, hybridize it with the DNA of another species, and see how much of the radioactivity is retained on the filter. Gives you a pretty rough idea of the overall degree of identity in the two genome sequences. But this is pretty rough. But you can get genus level and species level identification from that. If you want finer identification down to the subspecies level, you need more precise information. So this you will get from sequencing or from these genetic fingerprinting approaches with the repeated sequences in the genomic DNA. So with these types of approaches, you can classify species, genera, and strains within a species. For more high-level classification, then you want to study genes that are highly conserved, present in all bacteria, and that will remain stable over time. So perhaps the best is 16S or small subunit RNA. So this will enable you to do high-level classification. And also ribosomal protein sequences are useful for this as well. Okay, so then you can classify bacterial genera into families, phyla, and distinguish archaea, eukaryotes, and eubacteria. Okay, so this is a study on Neisseria again from a different team. 20 years later, the same group of bacteria. This time it's phylogeny based on sequencing of ribosomal subunit genes, RNA and proteins. And they also did whole genome sequencing on a number of their samples and found that, and they showed that their classification was not changed by taking into account more genes. So this whole genome sequencing thing is quite popular because it's possible to do it. So lots of teams want to do this, but it's maybe not necessary in all cases. It's like using a big tank to go and, I don't know, squash a pumpkin or something. You don't need such a huge tool to do that. Anyway, so here is their classification, and it's too small to read, but pretty much coherent with what was previously done with characteristics. This is from the first paper. I think there are some differences, but you have to look a bit closer. Let's see if I can zoom this in, because come back. Yeah, okay. Okay, so are we getting the same classification? Yeah, okay, so here you've got the two types of gonorrhea, they're clustering together. Same with the molecular classification. Then you've got the meningococc group here. They all group together. It's the same as here. Here is something different, okay? You've got the Neisseria meningitidis that's, or Cinerea that's in between these two groups of meningitidis. This is not correct, okay? It seems to be a mistake, because with molecular methods, the Cinerea group is really phylogenetically quite distant from meningitidis group, okay? So this is probably a truer reflection of the evolutionary relationships between these bacteria than this one, because you would have to look back in the data here and figure out exactly which characteristics were involved in placing this one this group of bacteria together with the meningitis bacteria, and figure out whether it's homeoplasia or not. Okay. So this was at a genus level. Okay, so within a genus, Neisseria, you can use molecular methods to construct phylogenies of the different species within the genus. Does it work at a higher level classification? Can you construct a phylogenetic tree that has all the bacteria that we know? So this is looking at that type of level here. So it's ribosomal subunit genes. It's difficult to see when it's projected, but here you've got an unrooted phylogram, and on each of these spots is one species of bacterium, one taxon on each of the kind of leaves. And what you can see is it's working pretty well, okay, because all the Bacillus species, they form a cluster. The Listeria species, they form a cluster. Staphylococcus, they form a cluster. Streptococcus, they form a cluster. Ah, but look, what's happening here? This is a bit wrong, isn't it? Got some Bacillus here, some Bacillus over there. So generally it's working, but there are some inconsistencies with what microbiologists would consider to be a correct classification of the, I guess these are the Bacillales, or yeah, class Bacilli of the phylum Firmicutes. Okay, so keep that in mind. Generally it works, but there are some inconsistencies. And we'll talk about why that might be later on. So that's within one kind of order. And this is all prokaryotes here, right? In a very, very complicated phylogram. So you've got, I have to zoom this in as well because it's too small to see really. Oh, okay, come back. Okay, one of the things I want you to think about here is gram-positive, gram-negative, all right? So bacilli, these are all gram-positives. They're all going together. Okay, that's okay, that's all right. Alpha, gamma, beta, proteobacteria, these are all gram-negatives. Actinobacteria, gram-positives. And you've got the roots, the deep roots around here. One of the things that's wrong here is you've got delta proteobacteria in amongst these gram-positives, Clostridia, Bacilli. Campylobacter is there, that's gram-negative. Helicobacter, these are gram-negatives. So you do wonder what are they doing around here, these guys? So once you get up to a higher level of classification, then you tend to get more and more inconsistencies. So that's with ribosomal protein sequences. With ribosomal RNA sequences, you can pretty much try and get the same result, that is, try and construct phylogenies of higher level bacterial taxonomy. And that's what it looks like. Okay, if you construct 16S or phylogenetic tree on the base of small subunit RNA sequences. This seems to be more coherent than the trees you get from looking at different collections of proteins. Why? Because, well, at least in this case, you know, you've got all the gram negatives clustering together and the gram positives clustering together. And you have the archaea out here as a kind of extra group. So you can see the root of your eubacterial tree is over here. Now, this is quite interesting because it might help us understand what was the lifestyle and the physical or the characteristics of the first types of bacteria, because the root is up here. One thing that is interesting is Acryphex spermatoga. These are extremophiles. They live in hot springs with a kind of chemolithotropic metabolism. So if this tree is really a correct reflection of history, then perhaps the most ancient bacteria had this kind of lifestyle. Another thing you can say is, okay, we've got gram positives here, gram negatives there. They are sister groups. What was the ancestral state? Were the first bacteria gram positive or gram negative? One cell membrane or two cell membranes on the outside? Okay, so that's where things start to become confusing. Chloroflexi, they're gram positive, okay. Deinococcus thermus, they seem to have two membranes, but they don't have LPS, so it's not a standard gram negative type membrane. Once you get up to here, Acryphex, clearly gram negative. But once you look in the genome, you can see that in fact they've got a lot of genes that have come from proteobacteria down here. So there's been lateral gene transfer of a lot of genetic material up into Acryphex. Spermatoga is also an interesting case because it's got a lot of genes that seem to have come from archaea. So it's a kind of mix of archaeal and eubacterial genes. And once you have this kind of thing going on, then it means that the taxonomic status of these bacterial species is very, very difficult to ascertain with certainty, because Spermatoga, it's up here near the root, but that might just be an artifact because it's got a lot of genes that have crossed over from archaea. So that means you can't be sure really about of anything to do with the origin of these species that are placed up here in the tree. Or can't be sure, or maybe can't be sure, okay? So that being said, one of the things that has come out of the study of short subunit RNA was the three-domain theory. This was a big surprise from results in the 1970s from Carl Woese, and which showed that what people generally considered as bacteria are not one homogeneous group, but there are eubacteria and archaebacteria. And the difference between archaeal ribosomal RNA and eubacterial ribosomal RNA is really quite significant. So that became the basis of this three-domain phylogenetic tree for the whole of life on the planet. So it's like this, okay? So eukaryotes are down here, bacteria there, archaea over here. Now there are a few consequences of taking this tree at face value. One of them is, well, they're up there on the slide there, is does the term prokaryote make any sense? we think of things in terms of prokaryote versus eukaryote. But this phylogenetic tree is telling us that that kind of bifurcation is not really a true reflection of the history of life on the planet. So some people, a minority of microbiologists, maybe a couple of people have said we should abandon this term prokaryote. It doesn't mean anything anymore. Another thing that seems to be indicated by this tree is that the eukaryote lineage must be very, very ancient. Why do I say that? Because in a phylogram, the branch length is correlated with the evolutionary distance between taxons and between nodes in the tree. And if all the groups are evolving at a constant rate, then these branch lengths should be correlated with time. So the branch lengths within bacteria are all pretty short. Within archaea, we're all pretty short. Within eukaryotes, they're all quite long. So that seems to indicate that this event, the origin of eukaryotes, is really very ancient. But there's no fossil evidence to support this claim. So this is a problem with this tree. And the other one is really the big question: does this tree represent the evolution of organisms? So it represents the evolution of ribosomes, in fact. Can you always say this is the same as the organism that carries the ribosome? Most people would say yes. And this, it might not be perfect, but it's the best we've got. Because I don't think, it's too difficult to tell whether lateral gene transfer will also touch ribosomal RNA, but it's a difficult hypothesis to test, but it's going to be the best kind of data we've got. So just coming back to the status of the eukaryote lineage, okay? This is the three-domain model of the history of life here, right? And one of the criticisms of this tree is that the way it looks is very highly dependent on the sampling. So we haven't really sampled a lot of archaebacteria, and the eukaryotes look like a completely different group. But maybe that's an artifact of just low sampling frequency of archaea. So there's been quite an effort to try and sample archaebacteria from different places and try and have a better phylogenetic coverage of this part of the tree. And this has really produced some very interesting results, all right? So this was published in Nature 2 years ago. And they found Loki archaeota, which live in an undersea volcanic vent in the North Sea. And if you add these Loki archaeota into, a phylogenetic tree, not just ribosomal RNA, but 36 conserved proteins. Here are different groups of archaea. Here's kind of like archaeota. These would be the ones that are perhaps used to produce this tree. And here, archaea seem to be grouping inside, eukaryotes seem to be grouping inside the rest of the archaea. What is this telling us? This is telling us that probably the three-domain idea about the origin of life is probably not accurate. And that better sampling of the archaea gives us a more accurate phylogenetic reconstruction. So probably this represents the history of the eukaryote lineage a bit better. Okay, eukaryotes cluster within one part of the archaea. Now, part of this story is the origin of genes that were specific for eukaryotes. So up until this paper, really there are a bunch of genes that are present in eukaryotes and not found either in eubacteria or in archaea. And it's kind of a mystery of where these genes came from. And these are genes for things like membrane transport within cells. Genes for proteins involved in membrane transport within cells. And there's a big surprise here. So here are a bunch of these eukaryote-specific genes. Okay, like this. And what is really quite nice is this group, the Loci Archaeota, have got a lot of these eukaryote-specific genes. So the specific functions that make us eukaryotes probably were not invented in the eukaryote lineage. They were invented before in the archaea, and then some of them seem to have been lost, okay, in the Loci Archaeota. So this was, I think, a pretty major result that resolves some of the questions about the origins of the eukaryote lineage. Okay, right, before I finish up on this subject, just to go back to a couple of things that I forgot to mention when I was talking about identification. So how do you identify bacterial species? Classically, it would be somebody would isolate a bacterium, look at its microscopic growth characteristics, oxygen relationships, bit of the biochemistry, and then say, oh, that's it, I've discovered Bacillus McIlroyii, and name the bacterial species after themselves or their hero, or their scientific hero or something. Then things were, there was a big attempt to put things on a more objective footing with Bergey's Manual of Systematic Bacteriology. So there were definitions to try and say that bacteria are the same species if these matching coefficients are greater than 0.8, or if 16S rRNA has greater than 90% homology. Of course, whenever you have this kind of numerical cutoff, it's like when somebody passes their exams or doesn't. Okay, if you say 10, that's the pass mark. You're always going to have somebody who's on 10.01 and somebody who's on 9.98. And so it's going to be like this with bacteria. You'll always have two bacterial isolates. One of them's got 90, if you compare them, they'll have 96.9% rRNA homology. Do you put them in the same species? It's a little bit arbitrary. So defining bacterial species is difficult. Part of the problem is disconnected from the rest of biology because in plant animal species, I think people use something like the biological species concept or the ecological definition of species, something that couples together the reproductive system and the ecological niche occupied by the species. That's not how it works with bacteria. So you have some kind of strange things like Shigella and Escherichia. If we go by the classification, these are supposed to be two different genera, but in molecular in terms of molecular phylogeny, they're very close, and they can exchange DNA with each other. So normally, you would say that these two should be the same species. And probably they are, but of course, this is for historical reasons. That these two isolates were, okay, two species were defined separately and have retained their old species names. And the last thing here is that one of the problems with bacterial phylogeny and really trying to interpret the deep roots of the bacterial phylogenetic tree is lateral gene transfer. So as we saw with Aquifex and Thermatoga, these bacterial families have undergone really a lot of gene transfer. And so some people say, some evolutionary biologists say that it's just a mistake to try and reconstruct a phylogenetic tree that goes all the way back to the origin of life here, with just two major events of lateral gene transfer. So first mitochondria into the eukaryote lineage, and then chloroplasts into the plant lineage here. So this guy, William Ford Doolittle, he published a kind of discussion article in Science almost 20 years ago saying that's what we teach in schools or in universities. And he said we shouldn't, because it's just incorrect, because the reality looks a little bit like this. You have a lot more lateral gene transfer than is represented here. And if this is the reality, what does that mean? It means we can never really get a clear lineage. It goes right down to the early steps of evolution of life on the planet. You can maybe get a classification up to around about here. Maybe from the origin of the eukaryotes about 1.5 billion years ago. But going back further in time, you just can't construct a tree, because, okay, you can construct a tree, but that tree won't mean anything. That's the message. And we're fooling ourselves if we think we can really reconstruct the history of bacterial life. So that is the end message, a little bit discouraging. Perhaps it's more of a, it's just a warning, okay? When you see things like this, we should always be careful about their interpretation. Okay, this kind of tree here. Okay, so I'm going to go on to nutrition and energy metabolism. So, yeah, we have about 2.5 lectures on this. Generally, I've done it in 2 lectures before, but I always ended up rushing towards the end as I wanted to. So, yeah, we've got about half an hour left, and the reason we talk about this is to explain this second point about bacteria. They can live on practically anything. Most of the types of energy metabolism, well, that we understand is going to be organisms like us. We eat organic carbon and we digest it. That's how we get our energy. Or plants. They photosynthesize and they get metabolic energy. So there are plenty of bacteria that work like that, but there are others who do very, very strange things. And that's interesting to understand. So what we're going to be doing this for the rest of the morning and tomorrow afternoon is firstly look at the basics of bacterial nutrition. Then just to make sure everyone is following this, probably tomorrow I'm going to start from here, talk about or give you a bit of a reminder about basic concepts in energy metabolism, and then we're going to really attack and try and understand the different types of bacterial metabolism. Going from things that are familiar to things that are really quite strange down here. Okay, so the next couple of slides are not fantastically exciting, but it is important to know this stuff. So every type of organism needs to get carbon, needs to get nitrogen, needs to get sulfur, needs to get phosphorus, needs to get, minerals. So what are the sources? It can be organic carbon, in which case the bacteria are called heterotrophs, and it can be inorganic carbon, so carbon dioxide, in which case our bacteria will be classified as autotrophs. They assimilate inorganic carbon and produce organic carbon, autotrophs. Source of nitrogen. Well, if you have amino acids hanging around, you can take them up and use them as a source of nitrogen. Even if all 20 amino acids are not present, most bacteria can transform one amino acid into another, or use biosynthetic pathways coming from other carbon compounds to transfer amine groups and produce their amino acids. Often ammonium, if it's free, can easily be incorporated into amino acids. So these are easy, okay? And you'll have ammonium present if bacteria live in a reducing environment, no oxygen. If bacteria are present in an oxygenated environment, probably there's not very much free ammonium around, but there might be nitrate. In which case, the first step towards assimilation will be reduction of the nitrate to ammonium, and then its incorporation into amino acids. And the most amazing one is nitrogen fixation. So reducing nitrogen gas into ammonium. I'll talk about this a little bit tomorrow when we look at the nitrogen cycle. Just now I'm just going to say this is extremely important for agriculture. Legumes, they fix nitrogen. The plants don't do it themselves. It's their symbiotic bacteria, rhizobia, that perform this function. Oh, what's going on now? Okay, phosphorus and sulphur. Why do you need it? You need it for producing phospholipids, ATP, DNA, and RNA. All of these can be sources of phosphorus for bacteria. If there's any dead animals, dead plants lying around, then their DNA, their RNA, their phospholipids can be used as a source of phosphorus. Inorganic phosphorus easily assimilated by many bacteria. For sulfur, okay, the amino acids methionine and cysteine can be used as a source of sulfur, or sulfate and sulfide. Once again, the form of sulfur that can be directly incorporated into organic molecules is the reduced form. So hydrogen sulfide will be directly incorporated into cysteine, whereas sulfate will first be reduced and then the hydrogen sulfide incorporated into cysteine. Okay, mineral salts. All bacteria are going to need large amounts of sodium and potassium for ionic balance inside the cell. Magnesium cofactor, mostly ATP. When it's used as an enzyme cofactor, it's not on its own. It's complex with a magnesium. So all organisms need magnesium, or everybody needs chloride to balance out the positive charges. And these oligoelements are essential cofactors for a lot of enzyme systems. Manganese, cobalt, iron, very important. A lot of redox reactions are catalyzed by enzymes that have got an iron-sulfur cluster in them. So iron is an essential growth factor for many bacteria. And finally, some bacteria can produce everything they need from basic subunits, but many will require some kind of growth factors. So thiamine, vitamin B1 and biotin B7 are required by quite a few bacteria. Well, what are these useful for? Biotin, it's essential for carbon metabolism, transferring one carbon groups. Thiamine is cofactor for transferring 2 carbon groups. And these are going to be essential parts of energy metabolism, this kind of carbon transfers. So if bacteria can't produce these vitamins themselves, they'll have to be assimilated from or provided in the growth medium. Now, one thing that is related to this is the concept of prototrophs and auxotrophs. Now, a prototroph can produce whatever it needs, but this term is always associated with a particular type of growth requirement. So if we're just thinking about these vitamins, these growth factors. Now imagine you're making up a very, very simple defined medium for the growth of bacteria, right? You put in carbon source, nitrogen source, and a few minerals. Okay, very simple defined medium. You'll have, I don't know, some glucose. You might have some ammonium salts, a little bit of inorganic phosphate, maybe some sulfate with sodium, potassium, magnesium, and chloride, and maybe a little bit of iron. If you put all these things into a tube and inoculate your bacteria, it might be able to grow. In which case, it is able to synthesize all of the amino acids, nucleotide bases, purine and pyrimidine bases, and all the growth factors. So that would mean this bacterium is prototroph for amino acids, purine and pyrimidine bases, and growth factors. Now imagine you inoculate your bacterium into this medium. It doesn't grow. But you add in thiamine and biotin, then it grows. So it means it's prototroph for all the amino acids, purine and pyrimidine bases, but it's auxotrophic for these ones. It needs these growth factors to be added. Okay? So when a bacterium is an auxotroph for, I don't know, purine biosynthesis, it means you must add this supplement into the basic defined medium. So starting with prototrophic bacterium and obtaining auxotrophic mutants was an important step in defining the biochemical pathways for the synthesis of all these different stuff in bacteria. Okay, because you could get a bunch of mutants, see whether they complement each other, how many genes are involved, this kind of thing. So this historically was very important in defining the biochemical pathways of amino acid, purine, pyrimidine biosynthesis, and so on. Now, when I talked about defined medium, I missed out some things. I did not talk about adding manganese and cobalt and so on. That's because these are generally trace contaminants in the salt that you use to make up the medium. So if you want your bacteria to grow in a minimal medium, you order the cheapest sodium chloride you can find in the catalog because it's not highly purified. So there will be trace amounts of other metals that the bacteria will need as cofactors. But if you are studying the growth requirements of bacteria, whether they need manganese, whether they need cobalt, then you must order the highly purified sodium chloride and make sure you don't have any of these trace contaminants. But generally speaking, you don't need to add these things specifically into the medium. Okay. So bacteria need to get all these things, and they need to assimilate them into the cell. So how do bacteria take up nutrients? Passive diffusion. Well, bacteria are pretty small, right? So they don't have to transport things over a large distance. But the only problem they have is getting them across the membrane, really. So passive diffusion, this will work just for oxygen, carbon dioxide, water. Facilitated diffusion. Okay, that means what? Just trying to see what I can use as a visual aid here. There we go. This is my bacterial cell. Facilitated diffusion works if you have a high concentration outside the cell and a low concentration inside the cell. But without any intervention, whatever is trying to diffuse into the cell cannot get in there. It's impermeable. So you need to have some kind of proteins that will allow whatever solute molecule is available to diffuse into the cell. So you need a hole in the membrane, which is going to be the permeases or porins. What is the difference? Porins are not very specific. So you find them in the outer membrane of gram-negative cells, and they will let in anything which is a small size and water soluble. Whereas permeases have a specificity for a substrate. Okay, facilitated diffusion will only work if there is a concentration gradient going from the outside to the inside. If you want to assimilate something that is at a very low concentration outside the cell, then you'll have to use some kind of active transport mechanism, which we're going to look at next. So the three types of active transport mechanisms you need to understand are ATP binding cassette transporters, ABC transporters, symport antiport systems, and group translocation. And we'll do iron at the end. So ABC transporters. These are in the plasma membrane, gram-positives and gram-negatives. They work with a solute binding protein, which is in the periplasm. So whatever you want to take up into the cell, going to fix first to the solute binding protein, then this complex binds to the transporter, and import of the solute requires ATP hydrolysis. So in E. coli, some sugars are taken up by this system, and many amino acids. Not glucose, okay? Glucose is group transport, as we'll see. But, rather unusual, more rarer sugars, maltose. But amino acids, think about that, glutamate, histidine, leucine. Symport antiport systems. Okay, in this case, the transport. So, okay, for ABC, let me just step back a little bit and say something quite simple about this. So active transport, it's what? It's importing something against the concentration gradient. So to get this to work, you have to put energy in somehow, because you're working against entropy, right? So, okay, for ABC transporters, the energy input is ATP hydrolysis. Okay, that's simple. For symport antiport systems, the energy input is going to come from a proton or sodium gradient that already exists across the metaplasma membrane. So, as part of their energy metabolism, bacteria have a proton gradient across the plasma membrane. So there's a higher proton concentration in the periplasm than in the cytosol. And this is one of the forms of metabolic energy in bacteria. So what you can do is co-transport sugar or amino acids with a proton or sodium import, because the energy cost of importing the solute against the concentration gradient is paid for by the positive energy contribution of this co-transported ion that's going to go back to the cytoplasm. So in E. coli, you can import lactose by proton symport or amino acids, but often this is the one to remember, okay? Malate oxaloacetate. These are 4-carbon molecules here. Sodium symport, amino acid glutamine. So for each one of these, what you need to remember is the diagram of how it works and one example in E. coli. Okay, so E. coli, ATP binding cassette, I don't know, glutamate, histidine. Symport system, proton symport, malate oxaloacetate. That would be the one to remember. If you're clever, so sodium symport, glutamate, this one will work as a correct answer for both of these systems. Okay, you can optimize your revision here. Group transport. Now this is the one we like to spend a lot of time on because it's just so clever. Me and Mr. Secagnon, we love it. We love group transport. Okay, so what is happening here? Now let me see if I can try and explain this in a sensible way. Okay, so this is the one for glucose, all right? Group transport E. coli glucose. Always remember that. So in the group transport system, you've got enzyme 1 and then enzyme 2. Enzyme 1 over here, what's its function? It's picking up a phosphate group from phosphoenol pyruvate that gets transformed into pyruvate. This is the end step of glycolysis. Normally in glycolysis, this would generate 1 molecule of ATP. But instead of generating 1 molecule of ATP, the phosphate group here is transferred onto enzyme 1. Then it goes on to another protein called HPR, heat stable protein. So you've got the transfer to the phosphate group, and then the phosphate group is going to be transferred onto the group 2, or the group 2 proteins. So group 1, E1 and HPR, these are conserved, they're the same, whatever sugar is being imported. Group 2 proteins, they're specific for each particular sugar that's imported, right? So group 2, first you've got a bit more of the phosphate transport, and then 2B and 2C, what they're going to do is allow the import of the glucose, and the energy from hydrolyzing this phosphoester bond here does 2 things. It's used to import the glucose against the concentration gradient and transfer the phosphate onto glucose. And why is this a useful thing to do? Because you might think, you know, it's complicated. You've got 1, 2, 3, 4, 5 different proteins involved and a whole complicated system. Why don't you just use an ABC transporter? You just need 2 proteins, one in the membrane, one a solute binding protein. It's much simpler. Well, in this case, if you imported glucose with an ABC transporter, you would need to pay 2 ATP molecules. One just to get your glucose inside the cell, and 1 ATP molecule to phosphorylate to get glucose 6-phosphate. With group transport, you couple these 2 processes because phosphorylation of glucose is the first step in glycolysis. You're going to use this glucose to get energy. So these 2 processes are coupled here, and it's a saving. You save the cell, and the bacterium saves 1 ATP molecule by doing this. Very, very useful. So that's why, group transport or a phosphotransferase system is common in many anaerobes. E. coli, Salmonella, Staphylococcus, and obligate anaerobes. But you don't find it in obligate aerobes, like, I don't know, Pseudomonas species. As we'll see tomorrow, these bacteria will be generating metabolic energy by fermentation. And fermentation does not generate very much ATP per glucose molecule. It's like 2, net gain of 2 ATP per glucose. So if you can save 1 ATP molecule per glucose, then that's really a big, it has a big impact on the energy metabolism of anaerobic bacteria. For aerobes, it's not such a big deal, okay? Because they're going to generate maybe 30 molecules of ATP per glucose, so they can spend 1 extra just to import the glucose. Hey, no big deal. Get a taxi, why not? Yeah, I'll pay. Okay, so that's what's important about these group transport sugar phosphotransferase systems. They allow bacteria to get their glucose cheaply, or transport it cheaply anyway. Okay, yeah, absent in most obligate aerobes. Ion transport. This is the last one, and then we'll stop for today. Okay, it's a problem for pretty much every species of bacterium is how to get iron, because it's an essential cofactor for all sorts of very important enzymes, and in an aerobic environment, iron is highly insoluble. So the concentrations of free iron that exist out in the ocean, for example, are going to be very, very low. So all bacteria need to have a specific transport system for iron. And this is also true for other metal ions. So for magnesium, this kind of stuff, you'll have a similar system. So E. coli, it's the model, but this is true for many bacteria, is they will secrete siderophores. So in E. coli, it's enterobactin, and enterobactin is, well, complex organic molecule that has the ability to complex, Fe3+. Well, my 3 is in the wrong place here, right? Should be higher. This is ferric or ferrous iron. Anybody remember that from chemistry classes? Way back when you were kids? Okay, like last year. Nobody remembers. Okay, I believe this is ferric iron. Iron 3 plus, I think, is ferric, and iron 2 plus is ferrous. Yeah, maybe, but we should all check this one, right? So anyway, the siderophore got a very, very high affinity for iron 3 plus, so it's going to form this complex, and the whole thing is going to be transported back into the cytosol via an ABC transporter. So this is playing the role of the solute binding protein. It's going to, okay, it's a small molecule, will be able to diffuse back across or through an outer membrane pore in, and then it will be imported by active transport by an ABC transporter because the concentration is very low. Now, this bond here is very, very stable. So, of course, it's no use for the bacterium just to have this complex inside the cell. It's got to release the iron. And in fact, the enterobactin has to be cleaved enzymatically to release the iron and then resynthesized for every atom that is imported. So it's a tricky system. Takes a lot of effort for the bacterium to get iron and other metals, but it's essential, really an essential import mechanism.